Hi guys, so welcome to a new series by Pureline Tutoring Service, and this one is called Exam Bootcamp CSEC Edition 2026, of course. Yes, I know the last time we were here was like a year or two ago, I know, but a lot of things was happening within that period of time. But the most important thing is that we're back, and we are back in the right time, which is exam season. So exam is quickly approaching, and as usual, we are here to help in whatever way that we can. So, with this new exam bootcamp series, what we want to focus on is actually getting down into the detail and incorporating some exam papers or exam questions, sorry, with that. So, what we'll be focusing on is letting you know the importance of that particular topic, talking a bit about some of the objectives, especially if it's the longer ones, the topics with the longer objectives. We'll just focus on the few that persons may have a bit of a problem remembering, or try our best to sort of summarize everything or the key points of that particular topic. We'll be looking at some common mistakes that are made with students. So, for example, mixing up definitions, mixing up labels, probably not giving sufficient examples or naming the required systems, whatever it may be, as well as ensuring that at the end of the session, we include either an entire past paper question or part of, based on the information that would have been given within that session. And we'll be looking at how to answer efficiently. So, for example, how to answer a question that is with 1 mark compared to one that is with 5 marks. There is a major difference that will allow you to get the full marks that CXC owes you. So, with that being said, I hope you enjoy this session. I hope it is useful. I hope you like, share, subscribe. Let your friends know that Pureline Tutoring Service is back. And this time we are back with the inclusion of human and social biology, as that was one of the topics that a lot of persons sort of requested or wanted to know if we had done. So at the end of the video, I'll give you a little more information about something else, but let's start with our first session. So for this first session, we want to start up with human and social biology, and we'll be looking at the digestive system. So under this, what we'll be doing today, we're going to talk about why the topic matters. Not in much detail, of course, because we're trying to keep the videos concise. We're going to talk about the organs that are associated with the digestive system, talk about the difference between the two types of digestion, which is mechanical and chemical digestion. Then we're going to talk about some common mistakes that are made when students are reviewing the topic, as well as finally finishing off with a past paper question. So here we have the organs associated with the digestive system, and we'll talk about what happens at each one, not in much detail, because remember this is just a quick summary that we're doing. But to start with, human beings, we do all our digestion on the inside of our body. So we have an internal digestive system. This muscular tube, which we call the alimentary canal, it starts at the mouth and it ends at the anus. So let's do a quick review or recap on what goes on at each part. So we start at the mouth. At the mouth, we have two processes taking place. When it comes to digestion, we know that we have two types of digestion. We have mechanical digestion and chemical digestion. Mechanical digestion, also known as mastication at this point, is where we use our teeth to help chew or grind the food, so breaking up large particles into smaller ones. Then we have chemical digestion, which includes the use of enzymes. And if you could recall, enzymes are biological catalysts, that speed up chemical reactions without being changed at the end. So they remain the same. They just break up the substrate into products. So the first enzyme that we meet in this whole process of digestion is amylase. And amylase starts working on carbohydrates, breaking down starch into maltose. So apart from that, the saliva serves the purpose of softening the food and putting the pieces together basically with the help of the tongue to form that circular structure which is called the bolus that gets shoved to the back of our throat and then makes its way down this tube which is called the esophagus. The esophagus leads to the stomach. At the stomach, this is the second point where we have both types, chemical and mechanical digestion. Mechanical digestion in the sense that the food bounces from wall to wall as the stomach muscle contracts and relaxes to mix with the acid or the digestive juices that are secreted by the cells that line the stomach. So here we have two types of enzymes. We have pepsin and renin, and we also have hydrochloric acid, which is produced in the stomach. The hydrochloric acid serves the purpose of helping to break down the food as well, but also it helps with the process of ensuring that if we have any bacteria that we brought down with that food, the bacteria wouldn't be able to survive at that pH, and therefore it helps in that sense of protecting our body, as well as some other reasons, which I hope you all know. So from there, after the food is digested further, it is now in a liquid form, which is called chyme. So that chyme is released bit by bit with the help of a sphincter muscle that you find at the end of the stomach. I forgot to mention the sphincter muscle that we find here at the esophagus, which controls how much food comes into the stomach at one point, as well as to make sure it's one way in most cases, right? You don't want the digestive juices just randomly coming up your esophagus. But apart from that, let's move on. So we have the chyme, which is released bit by bit from the stomach into now your small intestines. In your small intestines, it is broken up into three parts. We have the duodenum, the jejunum, and the ileum. We should know that at the duodenum, we have two things happening, two very important things. You have the end of digestion, meaning that the food is broken down into small enough particles that can now be the second process, absorption. So it can be absorbed into your blood from the lumen, which is the space within your small intestines. So here now we need a bit more help. And even though we know that the lumen of the small intestines is lined by these microvilli, and within them, we know we have the lacteal as well as blood vessels. And we should also know that the microvilli themselves, very close to their surface, they release enzymes that also help finish this whole process of digestion. But apart from that, we should be familiar with the accessory organs. So we have the liver as well as the pancreas. The liver creates a substance called bile, which is made from the breakdown of red blood cells, so that hemoglobin. And the bile is not stored in the liver, so it's just made in the liver, but it is stored in the gallbladder. And the gallbladder, via the bile duct, releases the bile into the small intestines. What the bile does is it helps with this process called emulsification, which is simply breaking down large fat globules, into smaller ones, so that enzymes that break down fats, which are called lipases, can come and work on them. Then we have the pancreas. What the pancreas does, it releases a multitude of enzymes that finish off or help with the completion of digestion. It also releases a base or alkali substance that is going to neutralize that hydrochloric acid that is coming with the chyme from the stomach. And reason being, we should be familiar with the fact that enzymes are specific to temperature as well as pH. So the pH that is necessary for these enzymes that are being released into the small intestines to work, it is not that of an acidic nature. So this is why it's very important for that base to be secreted to neutralize the environment, thus allowing the enzymes to work at their optimum, which means that they are working at their best. So once digestion ends, meaning that the carbohydrates are completely broken down to glucose, then we have the fats broken down into fatty acids and glycerol, and we have the proteins broken down into amino acids. Those units are small enough to cross the membrane and get into the blood, which will then be transported via the hepatic portal vein to the liver, where it will then package or change it into whatever it needs to and figure out basically what needs to be done with these substances. So this absorption takes place all throughout the small intestine. And then whatever is not absorbed makes its way into this part, which is our large intestines. While moving through the large intestines, salts and water is taken back based on the body's needs. And then at the end, you have all of the undigested material, as well as things like dead cells and any microorganisms that would have been there will now be stored in the rectum. And that will remain stored there until our body is ready to get rid of it through a process of egestion, which we should know is different from excretion. So egestion deals with the removal of undigested material from the digestive system through the anus. I hope nothing that was said there was new information to you. So now let's move on to talking about the difference between mechanical and chemical digestion, which is sometimes a very common mistake made. So mechanical digestion happens two places, which we spoke about as we were going through the diagram above. So at the mouth, the process of mastication, which is the grinding or the chewing of the teeth, not of the teeth, sorry, grinding or chewing of the food using the teeth. Then we have churning at the stomach, where the contracting and relaxing action of that stomach muscle causes this churning action, helping the food, the solid food, to mix with the digestive juices, therefore becoming that liquid substance which we call chyme. Chemical digestion has nothing to do with a physical process, but as it says, only deals with the chemical process. And when it comes to that, this is where we use enzymes. We went through the definition of what an enzyme is, and this occurs at the mouth, in the stomach, as well as the small intestines. Anywhere enzymes are released, this is where we have chemical digestion taking place. So for example, we have three categories of enzymes that we can break up the enzymes in. We have carbohydrases, which focus on breaking down carbohydrates. We have proteases, which focuses on breaking down proteins. And then we have lipases, which break down lipids. So an example of carbohydrates, we have amylase. We have two types of amylase. You have the amylase you will find in your mouth, and you also have the amylase that you will find in the small intestines. It should be noted that the amylase that you will find in your small intestines would never be the same amylase from your mouth, because if you recall, we have that middle point, which is the stomach, that produces hydrochloric acid that would cause the enzyme or that amylase coming from the mouth to become denatured because of the pH. So by the time it gets to the small intestines, it's non-functional. So it would be pancreatic amylase that is being released from the pancreas, of course, an accessory organ. That is the amylase that would be functional in the small intestines. You have proteases, which focuses on breaking down protein. So you have pepsin, which is found in the stomach. Pepsin is able to work at its optimum at or not at, but in the stomach with that hydrochloric acid. You also have trypsin found in the small intestines. Then you have lipases breaking down lipids, so breaking up lipids into fatty acid and glycerol. This goes into the lacteal, as we should know, not directly into the blood. And for example, this would be released by the pancreas. So now we'll focus on some common mistakes made. And I'll just touch on, I think, which are like the major ones. So we have mixing up bile with enzymes. Please note, bile is made from the removal, or not the removal, but the breaking down of red blood cells. So bile is made from that hemoglobin, which we know is that pigment responsible for holding on to, in another sense, the gases, so the oxygen and the carbon dioxide, which red blood cells are able to transport or bind to reversibly, meaning that it can bind it and then release it, unlike carbon monoxide. So we'll talk a bit more about that whenever we are doing the respiratory system. Bile is made in the liver. It is not stored in the liver, it is stored in the gallbladder. And it helps with only this process here of emulsification. So this emulsification breaking down the large fat globules into the smaller ones. We also have the help with the neutralization of that acid from the stomach. Next, we have enzymes. Enzymes are involved in chemical reactions. We have the different types that we spoke about above. Enzymes are very specific in terms of temperature and pH for it to work at its optimum. Enzymes are made of proteins. Enzymes are not made from the breakdown of any other type of cell, and the remnants of that is basically reshuffled or recycled. Enzymes are made purposefully in the sense that each enzyme or each different type of enzyme only works specifically on one type of substrate. So enzymes and bile, two totally different things. Then we have absorption and digestion. Both these processes take place, as we know, when we get to the small intestines. But digestion itself starts at the mouth. It ends at the small intestines. And to digest something means simply to break it down. So we're breaking down something from large pieces to smaller ones. That is what digestion is all about. Whereas absorption happens only after the food has been digested or broken down into particles that are small enough to cross from the lumen of the small intestines into the blood. So absorption occurs when the food is completely broken down, and it happens, or it starts actually, at the duodenum, which is the uppermost part of the small intestines, and it continues all the way through the small intestines. A next major mistake that is made is not naming any enzymes. And I completely understand this in a sense, because you want to be careful not to say the wrong thing, thinking that, okay, well, if I say the wrong thing, I'll lose the mark for it. But especially when it comes to those essay type questions where they may ask you to explain the entire process of digestion, starting or beginning at the mouth and ending at the anus, you cannot answer such high profile questions without giving the name of some of the enzymes. Nobody is saying that you have to list every single enzyme, but I would recommend naming the key enzymes. So, for example, you only have one enzyme in the mouth, which is amylase. You should definitely name that. You only have two in the stomach, which is the pepsin and renin. You should definitely name that, as those two could be comparative in the sense of speaking about pH and how the enzymes are specific to pH. We have two totally different pH settings here. Then, when you get to the small intestines, there's a very long list of enzymes that you can name, but you could just name a few. So, trypsin is easy to remember, lipase is easy to remember, sucrase, lactase, and there are many, many more. Very easy to remember. The easiest types of enzymes to remember is the ones that end with ASE. So, looking at lipase, we should know ASE, okay, so enzyme, lipase, lipids, right? So lipase breaks down lipids, sucrase breaks down sucrose, lactase breaks down lactose sugars. So you could look for the easy ones that you could remember simply by the name. Of course, like pepsin, renin, trypsin, those you just have to remember that they are enzymes. But it's very easy to remember some of them because of this ASE that you will get at the end. So finally, we have this past paper question from May-June 2025. So this entire question was worth 10 marks, and we would be able to answer it based on the information that we would have covered within this session. So question 6, figure 4 shows part of the alimentary canal. So A, name each of the parts labeled E, F, and G. Without even just focusing on E, F, and G, we should be able to name all of these parts. So E, you can look where it is ending up, or where is it going to. We should know that this J-shaped bag here is the stomach. So what leads to the stomach? That is the esophagus. F, it is this dotted part here that we are seeing, or not really dotted part, sorry, the dotted part is what is behind it. But this entire organ here, we should know because of its proximity, or even the fact that we have G attached to it, and this tube going into this other tube that isn't labeled. We should know that this is the liver. And G, we should know that attached to the liver here, we have the gallbladder. Then part B asks us to name the substance that is stored in G. We spoke about this. Bile is made in the liver but stored in the gallbladder. So you just put bile. You don't have to put the substance that is stored in G. All of that is wasting time. Just put the name that they are asking for. It is 1 mark. Then part 2, state 2 functions of the substance named in B1. So they are speaking about the bile. So what does the bile help with? 1, the emulsification, which is the process of breaking down large fat globules, into smaller ones. And then 2, the neutralization of stomach acid. So it helps with neutralizing that HCl, which is the hydrochloric acid that is coming with the chyme, to ensure that the enzymes found or released in the small intestines is able to work at their optimum. Then finally, we have C. So name 2 enzymes produced by the part labeled H. What is H? We should know, again, associated here. H would be the other accessory organ, which is the pancreas. And just by the shape alone, we should know that this is the pancreas. So we identify that it's the pancreas, and the substrate on which each of the enzymes work, which each of the enzymes act. So what are the substances or the substrates that these enzymes work on? So this is why I'm saying that you should try to remember some of the easy ones, and that way if a question comes up like this, you would be able to answer it sufficiently. So I just named these two. So amylase, it works on starch, and lipase, the substrate is fats or lipids. Again, 4 marks, 1 mark each. Do not write any long sentences. Just put exactly what the question is asking you for. So that concludes our first session of this new series. And if in fact you would be interested in joining a more structured live HSB revision before your exams, you can do so by clicking the link in the bio, which will allow you to register, and someone from Pureline Tutoring Service will reach out to you giving more information. So I hope it was useful. This would also be useful for students who are doing biology and integrated science, because all of the sciences would cover this much detail with the digestive system. So as I said before, like, share, subscribe, let your friends know that Pureline Tutoring is back, and we are here to help you with your exams.